Hello everyone, myself Leena Prabhakar Zogi from DRGCOP Malkapur. So in part 1 we were discuss about the few drugs, that is the industrial production, estimations of and utilizations of the few drugs. Now in this part second we will discuss about another few drugs, industrial production as well as estimation and uses or utilizations of another drugs. So in that part second we will discuss about atropine. So what is the biological source of atropine? That is the tropin alkaloids. Then flowering tops of the Atropa belladonna, Datura stramonium and Hyoscyamus niger belonging to family Solanaceae. In industrial production, first step is it is the drug should be, it is in powdered form. So powdered drug should be extracted with ether or the benzene. Then in second step we have to concentrate the non-polar extract and partition with the acetic acid. In third step we have to add sodium bicarbonate, which is leading to precipitate the alkaloids. And in last step we have to dry the precipitate and the crystallize by dissolving in solvent ether. Then estimation, which is done by the TLC. In the TLC, stationary phase is the silica gel G and mobile phase is the ammonia is to methanol in 5:100 ratio. Then spraying reagent, it is the iodoplatinate solution and RF value should be 0.18. Another estimation, it is done by using the assay. So in assay we have to sulfate salt of the atropine titrated against 0.1 normal per chloric acid. Then utilization, atropine, it is used as a pre-anesthetic medication as well as it is also used as a antispasmodic drug. Then next drug is the podophylotoxin. So biological source of the podophylotoxin is the resin, roots and rhizomes of the Podophyllum hexandrum, Podophyllum imodi, Podophyllum peltatum belonging to family Barbadiaceae. So here is the structure of the podophylotoxin. Industrial production, we have to take the dried root or the rhizome of the podophylotoxins, which is extracted with the methanol, then evaporate the filtrate to semi-solid mass. In next step we have to dissolve in the acidic water, which is result in the precipitations of the podophylotoxin. Estimation, which is done by the HPLC, in which mobile phase is the methanol is to water in 62 is to 38 volume by volume. Detector wavelength, that is 280 nanometer. Then utilization, it is used as anti-tumor agent. Then also it is used as a purgative, emetic, and it is also used in the treatment of the warts. Next drug is the caffeine. So biological source of the caffeine is the xanthine alkaloid and it is obtained from the leaves of the Camellia sinensis, Thea sinensis belonging to family Theaceae, as well as it is obtained from the seeds of the Coffea arabica belonging to family Rubiaceae. Production, it is that means industrial production, that means isolation of this caffeine. So in this in first step we have to take the leaflet powders, which is boiled with the 2% sodium carbonate solution in the water for the 10 minutes and filtered. Then evaporate and partition with the dichloromethane in separating funnel. Then next step we have to evaporate to get the crystal of the caffeine and purified by recrystallization from the hot ethanol. Estimation, which is done by the HPLC method, in which mobile phase is the methanol is to acetonitrile in 65:35. Column C18 can be used for the estimation purpose or HPLC method. Utilization, it is used as a stimulant, CNS stimulant. Next drug is the Taxol. Biological source of the Taxol, it is nitrogen containing substance, that is the bark of Taxus brevifolia or Taxus baccata belonging to family Taxaceae. This is the structure of the Taxol and here is the plant of the Taxol. So powdered, in this extraction is which is done with the help of the powdered drug. Powdered bark can be extracted with the methanol and filtered and then evaporated to the dryness. That means we have to concentrate particular extract. In second step we have to partition these extract with the mixture of the carbon tetrachloride and water, then again filter and evaporate. Then again we have to take the dried CCl4 fractions, again extracted with the carbon tetrachloride is to methanol and evaporate to obtain the crude Taxol. Estimation which is done by the HPTLC method, in which mobile phase is the chloroform is to methanol in 7:1 ratio, and spraying reagent or the visualizing agent, that is the vanillin sulfuric acid. Utilization: Taxol can be used in the treatment of the ovarian, lung, bladder, esophageal and other types of the cancer. It is also used as an anti-proliferative agent. Next drug is the vincristine and the vinblastine. Biological source of the vincristine and vinblastine, it is the indole alkaloid which is obtained from the whole plant of the Vinca rosea or Catharanthus roseus belonging to family Apocynaceae. This is two structure of the vincristine as well as vinblastine. Production which is obtained from the plant tissue culture technique. So there are some uh, writers or the literature review from which the production can be taken here. So production which is obtained, vincristine or vinblastine, it is obtained from the plant tissue culture technique. Estimation which is done by the HPLC method, in which mobile phase is the acetonitrile, that is 0.1 molar phosphate buffer, and wavelength that is 254 nanometer. Utilization: it is used in the chemotherapy regimens, childhood leukemia and immunosuppressant. So thank you. This is some reference of this PPT, that is the Pharmacognosy and Phytochemistry Volume 1 4th editions by Vinod D. Rangari.